And now we will go for our last talk of the session with Kate Jeffery from University College London in the UK. And she will talk about egocentric versus allocentric neural coding in retrospinal cortex. So, stage is yours.
Thank you. I hope you can all see that.
Yes, perfect.
So I want to talk about something I've been interested in for a long time, and I want to talk a little bit about some of the work that we've done in the past to introduce some of the work that we've been doing more recently. Looking at this question that we've already addressed in this meeting about how the brain and the head direction system is trying to put together the information that's coming in from the outside world and assemble that into kind of a stable input for orientation. So we have already heard about the ring attractor. This is just a kind of a little animation that I made to show the dynamic nature of the problem. So that as the animal is moving around quite fast, the head direction system is having to update based on the self-motion cues. But the orientation of that entire system needs to be linked to the outside world in a stable way for the animal to be able to act in the outside world. So there's kind of two things going on. There's the internal dynamics, and how do you make sure that activity is happening in the right place in that ring? And then there's also the anchoring that to the outside world. And we've known for a long time, it makes perfect sense that the sense of direction is using information from the things in the world that are stable, and we call those landmarks. But actually, we don't really have a good characterization of what makes a landmark. So usually in an experiment, we'll be using some salient visual cue, like a piece of card or something like that. We assume that the kind of objects in the world are landmarks, but we haven't really dug into exactly what it is that the head direction cells use. And I think that's kind of an open question at the moment. And it's a question that's kind of raised by some of the results that I'm going to tell you about. So then there needs to be some kind of plasticity between the things in the outside world and the ring attractor, so that if the dynamic of the ring attractor becomes a little bit misaligned, so in other words, the animal becomes slightly disoriented, then perception of the familiar stable cues can retrieve the correct activation of the ring, so drive the bump of activity to be correct. So I started looking at this a very long time ago when I was a postdoc in John O'Keefe's lab, and I started kind of looking at what happens if you manipulate the relationship between the internal sense of direction and the environmental cues. And this is an example from an experiment that I did where I was recording in an enclosed environment where there were no particularly strong directional cues except for a cue card. And the animal's internal sense of direction could be manipulated by enclosing it so it couldn't see anything and then rotating it very, very slowly. And what I did was then do some kind of conflict trials looking at if you take the rat out, move the cue card somewhere else and put the rat back so that there's a conflict between the internal sense of direction and cue card, do the place cells, so this was place cells, not head direction cells, but do the place cells, does the spatial system go with the visual cues, or does it stay with the internal sense of direction? And what I found was that if the animal can see that the cue card is unstable, so if it's allowed to see that the cue card is moving, then over time, over subsequent trials, the percentage of trials in which the cells are reoriented by the card falls, and the cells become progressively more oriented just by the internal sense of direction of the rat. So the system started out trusting the visual cues, but after experience of their instability, it came to mistrust them, if you like. So there's an ability of the spatial system to learn about the reliability of the visual world. And that seems perfectly adaptive when you think about it, because the system doesn't come into being knowing which are the stable cues and which are the unstable cues in the environment. It has to kind of learn. And it's kind of a bootstrapping problem, because the system is also trying to use the landmarks to stabilize its internal sense of direction. So on the one hand, you're looking at the world and going, what are the cues? I'm going to use that to set my sense of direction. And at the same time, you're using the sense of direction and saying, which are the stable cues. So there's some kind of interaction going on. And I've been kind of curious about how and where that might be happening. So we started investigating retrosplenial cortex a few years ago and doing some experiments to look partly at how it's processing visual cues and also what's happening if you place the visual cues in conflict with the internal sense of direction. So this is an experiment that was done by Lave Lozano a few years ago, just looking to see whether these cortical head direction cells, which are very close to the visual cortex, whether they have the capacity to kind of distinguish fine-grained aspects of cues. In other words, like can they see the fine details of landmarks? And I mean, the point really, so what he did was had a symmetric environment with two cue cards at 180 degrees apart from each other. And one of the cue cards had one of these patterns of these pairs, and the other cue card had the other pattern. And the question was, would the cells always reorient the same way every time the animal went back into the environment? Or would it be sometimes one way and sometimes the other way? And if that were the case, that would mean that the cells could kind of see that there was a card, but couldn't really see the detail on the card. And some of the cards were really obviously different. So here, very high level of discriminability. And sometimes it was quite subtle, so a very low level of discriminability. And got the slide there. So basically, what we found was that that both retrosplenial and postsubicular head direction cells could orient very easily using the highly discriminable ones, and they could orient reasonably well using the more moderately discriminable ones. That's these ones here, and the ones that were identical, they were at chance. So that kind of tells us that there's some visual processing going on, although it doesn't tell us where it is. But that's really just to make the point that that vision is important for the system. The other thing about these cortical head direction cells is that if you look at the kind of the temporal characteristics of their firing, you find that the retrosplenial cells fire slightly in advance of the postsubicular cells. So without going into this in too much detail, if you look at how much the firing is anticipating the head direction of the rat. So this is a phenomenon that was discovered some time ago, that in some parts of this circuit, the head direction cell fires maximally not to where the rat's head is actually pointing, but to where it's going to be pointing in a moment. So that suggests that there's some type of predictive encoding, if you like, going on. And you can see on the right here that the anticipatory firing is higher for the retrosplenial cells than for the postsubicular cells. You can also see that in the fact that that this band here is sloped. So that suggests that maybe the retrosplenial cells are kind of driving the rest of the cells in that location. So again, it's only a hypothesis, it's not sort of proved yet, but there's also other reasons to think that retrosplenial head direction cells are particularly involved in the landmark processing part of the circuit. So studies in humans that have been done by Eleanor McGuire have found that the retrosplenial area in humans seems to be very, well, it's activated by lots and lots of spatial things, but particularly when some type of landmark processing is involved. And in a really nice set of experiments, she showed that people vary in how much they process the stability of landmarks, and that variance correlates with how much their retrosplenial cortex becomes active when they're processing the landmark stability, and also correlates with how good they are at using landmarks to reorient. So we sort of had a few reasons to think that retrosplenial cortex might be involved in processing of landmarks. And we decided to kind of dig into this in more detail a few years ago, using a sort of standard paradigm that we usually use for head direction cells, which is to record as a rat is foraging in a box. So normally there'd be a single container with a cue card on one part of it, like I said. And what we wanted to do was to kind of place a conflict, a kind of a controlled conflict between the landmark and the sense of direction. And the original motivation for doing this experiment was that we wanted to ask a question that I'd been asking for a long time with place cells and grid cells, which is, are the cells sensitive to the non-spatial context cues that are in the environment? So with place cells, if you change the odor of a box, for example, from lemon smelling to vanilla smelling or something like that, rats are very, very olfactory. And if you make a change like that, you'll typically get remapping. So the place cells will reorganize, sort of suggesting that the cells can use context cue to tell them which of the spatial cues, in other words, the walls and the kind of spatial structural parts of the environment, which subset of those should they be using. It's sort of a gating mechanism where the context cue is switching in the spatial cues. So we decided to do this with head direction cells. We already knew that if you just changed the box from lemon to vanilla, then the head direction cells wouldn't remap because they are very stable across contexts. So what we did was said, what happens if we have a context-dependent contingency between the landmark and the kind of global environmental direction, so that for the head direction cell to be able to use the environment to reorient, it would also have to use the context cue. So it would have to integrate the olfactory information with the visual information. So what we did was we created this two-box environment that has two rectangular subcompartments and a doorway, and the rat can kind of move in and out between one door and the other. And one of the compartments was centered with lemon and the other compartment was centered with vanilla. So this is where the kind of context dependence comes in. And in the lemon compartment, the cue card was at the, let's say, the north end of the box. And in the vanilla compartment, the cue card was at the south end of the box. And the question was just really, can the head direction cells maintain the same orientation in both of those environments, even if you close the doorway and just randomly put the rat in one or other? So a head direction cell that wants to fire when the rat faces north should fire, that way, regardless of which box it was in. Even though the visual appearance is rotated, the odor information, in theory, if this integration is possible, should be enough for the head direction cells to disambiguate those two situations. And we found that that was indeed the case. So if you look at the polar plot tuning curves of this really beautiful head direction cell, when you rotate the entire two-box compartment, then the cell rotates as you would expect. It was just to kind of confirm that that would happen. And then if you close the doorway and just randomly place the rat in one or other of these compartments, then you can see that the head direction cell fired in the correct direction for the odor. So in other words, it was able to fire one way with respect to the landmark in lemon and the other way with respect to the landmark in vanilla. So it could use the lemon or vanilla to retrieve the correct kind of orientation. So that was the kind of basic point of that experiment. And then we were kind of meant to move on and do something else. But we also actually discovered some other odd things, which is that mixed in with that population of retrospinal head direction cells, there were a subpopulation that had two tuning curves. So they had this kind of bidirectional pattern. And when we looked at what was happening in single compartments, we found that there was a singular tuning curve, like a normal head direction cell, but actually it rotated between one compartment and the next. So it looked as though these cells were ignoring the olfactory cue, not using it to disambiguate the visual situation. These cells were just reliant on the visual layout of the environment. And interestingly, we found that these cells were coexisted with the head direction cells. So when we do the manipulation, let the rat walk from one compartment to the other, that rotates the bidirectional cell, makes it fire in the other direction, it became dissociated from the head direction cell. So you can see that in this pair of trials here. So the head direction cell has maintained a global firing direction, whereas this other cell that looked like a head direction cell has flipped from one side to the other. So we seem to have kind of broken open this ring attractor. And it made us kind of realize that there were at least two populations of cells, and we started doing a lot of head scratching over why this might be. So we at that stage, we called them bidirectional cells. We called them bidirectional head direction cells. And they were mostly found in the dysgranular part of the retrospinal cortex, which is the kind of more dorsal part in the rat. Head direction cells, the ones that were globally stable, were found across both subregions. We also noticed some other odd things about these cells. So although it was true that a lot of these cells, if you just recorded in a single compartment, had a singular tuning curve, so they kind of look like head direction cells. When we looked a bit more closely, we found that that was only true for some of the cells, about 50%. There was another quite sizable population that actually had two peaks, even in a single compartment. And we tried very, very hard to break this apart and to show that, maybe we were accidentally recording two cells, and we did lots and lots of analysis to show that it wasn't the case. It's really a single cell that has two tuning curves in opposite directions. And, we'd been recording retrospinal cells for years and never seen anything like this. So somehow it seemed like it had to be that this bidirectional pattern has arisen from the rat's experience of these two compartments with the dissociated relationship between the visual scene and the head direction. It had to be some type of experience-dependent effect, we sort of thought. We also found that usually, although it was kind of bidirectional, there was usually one peak that was larger than the other, and that would typically rotate when the rat went from one compartment to another. So there was some information coming into that cell. Even though it's got the two tuning curves, it was also able to sort of distinguish one compartment from another. So we did a lot of thinking about how this could all be explained, and we concluded that what these cells are doing is they're sitting in retrospinal cortex. They're getting a lot of information coming in from the visual cortex about what the rat can see. So the layout of the environment. They're also getting a lot of input from the head direction system, which, is this big network that includes thalamus and sorts of other regions. So I call that the consensus signal, if you like, what the brain as a whole has decided as your facing direction. So these cells are getting these two sets of inputs. So the cells that had a singular tuning curve that rotated when the rat stepped through the doorway from one side to another, these were cells that seemed to be really strongly driven by the landmarks. So we imagined that these were cells that have strong inputs from the landmarks, and they don't really care what the rest of the head direction system thinks. So they're kind of mainly landmark-driven cells. Head direction cells are kind of the opposite. The head direction cells are expressing the decision that the entire network has made about facing direction, and they don't really care what the rat can see. So if there's a bit of a conflict, they don't care. They're still going to maintain their global orientation. And then these other ones, these within compartment bidirectional cells, the ones with the two tuning curves, even in a single compartment, we think that they have picked up accidentally, if you like, inputs from two sets of head direction cells. And an example that I just showed you from, let's say, the north head direction cells and the south head direction cells. And the reason that that's happened is because of this weird situation where the cell is being driven to firing threshold by the landmarks. And in one compartment, the landmarks coincide with one set of active head direction cells. And in the other compartment, the landmarks coincide with the other set of head direction cells. So they've kind of acquired two sets of head direction input, and that bolsters the signal and means that the cell will fire in either of those directions. So that was our kind of conclusion, or at least kind of hypothesis. So we decided to sort of pursue this a bit further because we had only ever tested the two compartment box where there was 180-degree dissociation between the layouts of these environments. And at around about the same time, some other kind of evidence was coming along from Doug Nitz's lab, recording from some other brain areas, in particular sort of subiculum, finding that there are cells that have 180-degree opposing tuning depending on which way the rat is running in a kind of a complex maze. So he called these axis cells, and they seem to kind of be interested in 180 degrees. And we sort of started to think, maybe it's the case that actually the part of the brain kind of cares about the axis of movement and, you know, the sort of forwards and backwards aspect of your trajectory, because in real life, most trajectories are quite linear. So rats, for example, will typically take up linear trajectories through the environment. So we thought maybe these aren't really head direction cells, that maybe actually their function is more of a kind of a route related one. So the simplest thing to do was to see if that 180 degree symmetry persisted if we change the symmetry of the environment. So Ningyu Zhang, who's a PhD student in my lab, did a comparison of what these cells would do in the original two-compartment environment, which we call the two box, in a single compartment, so the kind of standard ordinary kind of open field arena where there's just kind of a one-fold symmetry, if you like. And then a new box, which we call the four box, which is like the two box, except that there are four compartments and there are four cue cards. Again, there's a sort of a doorway in the middle, so the rat can walk from one compartment through the doorway and into another compartment. And in every compartment, as with the two box, the visual layout is the same. So there's a cue card on one wall, and then on the wall to the right of the cue card, there's the doorway. So that's true for every one of these compartments. So if you just teleported the rat into one of those compartments, it wouldn't know which one it was in because they all look the same. If the rat didn't know anything else, so if it didn't know anything about the outside world. So this now has a kind of a fourfold rotation symmetry. And then just to break the symmetry so that the system had something to hold on to, if you like, we appealed to the discovery that we'd made that the system can use the olfactory cues to break the symmetry. So at least the head direction cells can. So we centered two of these boxes with vanilla and two of them with lemon. So now if the cells know about the odor as well as the visual layout, there's now a unique orientation for these compartments. So for example, there's only one compartment in which there's vanilla with lemon to the right if you're facing the cue card. So if you know about the odor as well as what you can see, then you can work out which compartment you're in, if you like. So this was to help the head direction cells. And then the question is, what would the quote unquote bidirectional cells do? So we sort of expected this, but we were happy to find it anyway, that indeed in the four compartment environment, we now see what we call tetradirectional firing. So this cloverleaf pattern, where if you look at the pattern across the entire environment, there are four tuning curves. And if you look within a compartment, actually, I think there's a better picture here. If you look within a compartment, then there are these two patterns. There are the cells that produce just a singular tuning curve. So that fourfold pattern only arises when you consider all four compartments together. So these are the between compartment cells, which are analogous to the between compartment bidirectional cells that I told you about before. But then there's also this other population of within compartment tetradirectional cells. So even within each of these subcompartments, there's this kind of cloverleaf pattern. And again, we've never seen anything like this in an ordinary environment. And we didn't see any of the bidirectional pattern in the cells that were recorded in the four box. So these were recorded, and they weren't recorded in both compartments because we didn't want to mess up whatever plasticity was going on. So it's different rats and different cells in the two box and the four box. But we didn't see any bidirectional pattern in the four box. And of course, we hadn't seen any four directional pattern in the two box. So this kind of supports this idea that these cells are plastic, and that they're picking up some type of symmetry from the environment, from the experience of that environment. Then you then also looked at what happened in the baseline trials. So for every kind of sequence of multi-compartment trials, there was at the beginning and the end, there was a trial in the open field arena. And there was sort of a mixture of patterns. So there were some cells that really seemed to have no directionality whatsoever. So these two here, this is two cells. So this is one cell on the top two, and this is the second cell, the bottom two. And these are cells that in the two box had a bidirectional pattern. So these cells are capable of expressing multiple tuning curves. The top cell really didn't have any directionality at all in the open field arena. The bottom cell had a bit of one, so there's a bit of an asymmetry. There's a slight tendency to fire more in one side than the other. And similarly with the cells that were tetradirectional in the 4 box, here is a cell that didn't really show any directionality at all in the 1 box. And here's a different cell that had a weak kind of directionality. So the propensity to be directional is sometimes there and sometimes not in the 1 box. But it seems that it's something that's learned on experience in the 2 box and the 4 box. This is just to show you again in the 1 box. This is just a heat plot to show you the symmetry slightly more clearly. So there is definitely this slight sense of one-fold symmetry. There's definitely a if you take the tuning curve and map it onto itself, there's a variability in how much. Sorry, I think it's compared across trials. There's a variability in how strong that central peak is. In other words, how kind of one-fold symmetric the firing is of that cell, but basically it's pretty weak. So these don't really seem to kind of be classic head direction cells in very many ways. Again, these were mostly the multi-directional cells, and mostly found in the dysgranular rather than the granular part of the rich sphenial cortex. We also looked at the electrophysiological characteristics and found that the bidirectional and the tetradirectional cells were indistinguishable as we expected, because we think they're the same type of cell, but they were quite distinct from the head direction cells. So the head direction cells were kind of burstier, and they had they were narrower, they had a kind of a shorter peak to trough latency. So you can see from the waveforms, the head direction cells are quite narrow and pointy, whereas these other cells are much broader. So they seem to be a distinct population of cells by several different measures. So then we sort of turned to this question that had been kind of nagging us ever since 2020. So actually, since the last INAV meeting, when at least the in-person one where we last all got together, which I fondly look back on, look forward to happening again someday. So there was a sudden eruption of people reporting egocentric boundary coding in all sorts of brain regions and all sorts of settings. So egocentric boundary coding refers to the propensity for a cell to fire when there's a boundary that's at a particular distance and direction from the animal. And it still takes me a long time to kind of get my head around this, because it requires a kind of a shifting of reference frames. But for something like a place cell fires when the rat goes into a particular part of allocentric space. So when the animal goes into the place, then the place cell fires. For an egocentric cell, the reference frame is not the place but the animal itself. And the firing of the cell happens when the allocentric world comes into the space of the rat, if you like. So in other words, when a boundary comes into the sphere of influence of the rat, or its sort of zone of detection, or whatever you might like to call it. So for example, you might have a boundary vector cell that becomes active if the rat detects that there's a boundary to its left, and at whatever that is, 135 degrees or whatever. So these boundary vector cells are egocentric because they care not about where the rat is in the world, but about what the world is doing relative to the rat. And quite a few of these have been reported in this really nice paper by Alexander et al. In retrosphenial cortex. And if you look at the firing patterns, there are sort of some interesting kind of features of them. So they've been color coded according to head direction, facing direction. And you can see that you get these kind of very symmetric patterns, these kind of, for example, a fourfold symmetric pattern in this fourfold symmetric environment. And if you plot the hotspot in egocentric coordinates of where the cell is active, there's a very kind of focal point. So this here is the egocentric reference frame of the rat, and the hotspot is where in that reference frame, if there's a boundary, that cell will become active. So you can see that it makes these kind of nice hotspots. So it was sort of suggested that maybe our bidirectional cells were expressing a type of egocentric boundary coding, so that the tuning was actually arising from the structure of the environment, which was placing constraints on the experience that the rat had of their boundaries, of there being boundaries at a particular egocentric direction. So that for various reasons, we don't think that's the case. So one thing is, we thought if there's any, if it's something to do with the environment, so for example, if the 2-box is making the rat oversample some directions, then we should see an oversampling in the distribution of the preferred firing directions of these cells that kind of follows the symmetry of the environment. And we really haven't seen that for any kinds of cells. So if you plot the preferred firing directions of all of those tuning curves for head direction cells, for the bidirectional pattern cells in the 2-box, or the tetradirectional pattern cells in the 4-box, it's pretty much distributed around all the sort of compass points. So there doesn't seem to be an effect of environment. When we look at the kind of the color-coded, the directionally color-coded spike plot, so in other words, if you color-code each spike by the direction that the rat was facing at the moment the spike fired, we don't see any fourfold symmetry, which we would expect either within compartment. So because each subcompartment itself has a fourfold symmetry, we might expect a fourfold symmetry in each subcompartment. And also because the global box has a fourfold symmetry, we might also expect some type of global fourfold symmetry. Haven't really seen anything like that. And when we do the quantitative analysis, so we do those kind of hotspot plots of egocentric boundary coding, we don't see hotspots. I think these may be some of the hottest spots we saw, but you can see that they're kind of rubbish compared with the ones from the Alexandra et al. Paper. So when we kind of count up how many cells cross the threshold for egocentric boundary coding, we find that the vast majority of the cells that have the symmetric coding, you know, the twofold or fourfold patterns, don't have any egocentric boundary coding by our quantitative measure at all. So we don't think this is egocentric boundary coding. We think that these are really cells that are coding direction. So they're producing tuning curves based on the visual layout of the environment, or something structural about the layout of the environment. It doesn't have to be visual.
You have 2-3 minutes left before we have time for discussion. Thanks.
Okay, thanks. I've just got a couple more slides. So just to get back to how I started all of this, this seems to be a plausible means for the head direction system to learn about landmark stability. So what we have here are cells that are sensitive to the visual scene. We have cells that are insensitive to the visual scene and only care about the global consensus facing direction. So those are the head direction cells. And then we've got these kind of cells in the middle that are plastic, and we think that they're plastic because they're picking up connections from visual cues and from the head direction cells. So what we think that they're doing is looking for Hebbian conjunctions between the head direction signal and the visual scene that the rat can see. So for example, a landmark. So when there's a consistent conjunction, so every time the rat is facing north, it can see that landmark. And that's always the case. Every time it sees the landmark, the north head direction cells are firing, and every time the north head direction cells are firing, if the rat opens its eyes, it can see the landmark. So there's a really stable relationship between those things, and so Hebbian processes will cause that landmark to be added to the panoply of cues that can drive the head direction signal. If, on the other hand, there is a weaker connection, so for example, sometimes when the north head direction cells are active, the rat can see a landmark and sometimes it can't, that disjunction weakens those connections, and that cue becomes weakened and less able to drive the head direction system. And you can imagine that this is going on millions of times for all the many things that can be seen in the environment. And you'll be left, after this process has worked its way through, with just the most stable and the most salient landmarks being able to drive the ring attractor. So we did wonder about one other possible crazy function of this system, and this is extremely hand-wavingly speculative, and I wouldn't want to defend it strongly, but it's kind of interesting that the global symmetry of the environment is reflected in the local symmetry of the cell. So these within compartment cells that learn the global symmetry and then pick that up in their firing patterns. Now, it might just be an artifact. It might just be because we put the rat in this really artificial situation where there are all of these identical landmarks, identical environments, but they were all oriented differently. So maybe we've just messed the system up. But maybe, it has a function because the rat can now access information about the global environmental symmetry by looking at these cells. Might it ever do that in real life? Well, who knows? But rats do have these quite complex kind of environments that they live in and they create, and maybe it is useful for a cognitive mapping. In any case, as far as I know, it's the first kind of report of a global environmental structural signal being visible at the single cell level. So I think it's kind of interesting. But yeah, as I say, it might also be an artifact. So I just want to leave you with this conclusion. This is my last slide. This is kind of what we think is happening. So we've got these, I don't know what to call them now, multi-directional cells, landmark modulated head direction cells, whatever we're going to call them. I'll entertain ideas. And they're driven by visual cortex, and they're driven by the head direction system. And there's a lot of kind of interconnections, including with the non-spatial context and so on. This is all informing the place cells, and the place cells in turn are sort of feeding back, and it's all very, very complicated. But this area of retrosplenial cortex is where much of the plasticity happens that enables the integration between the environment and the internal sense of direction. So I'll leave it there, and thank you for your attention. Happy to take questions.
Okay. Perfect. Thank you very much. Really nice talk. And we already have like a question on from Kevin Allen. We say, nice talk as always. If the firing of bidirectional cell comes from the pairing of a north head direction and visual feature cells, would we not expect that the bidirectional cells would be influenced by the egocentric angle to this visual cue more than pure head direction cells?
I'm not sure quite what is meant by egocentric angle, but yes, it's true that the cell is more influenced by the layout. So the caveat about this is that because we've put a cue card there, everybody always thinks the cue card is the input to this system. We don't know that. It might be the entire panorama. So it might be that the entire visual field is processed by these cells. But yes, there's an egocentric component to that, because so there's a local component to that. So when the rat steps through that doorway, the local layout grabs control of those cells. I guess it's not really egocentric in that sense, because that cell still has that tuning curve in that compartment, no matter which way around the rat is facing. So I guess I would say local, but not necessarily egocentric. I'm not sure if we answered that properly. But
Okay, we have another question from Nicolas Schuck. Great talk. Do you see any evidence that multidirectional, either binary or tetra, firing pattern emerged later after more experience than any of unidirectional direction cells?
No, that was one of the things that we wanted to try and catch with this experiment, because when in the bidirectional cell experiment, we didn't know that we were going to find these, and so all of our rats, by the time we realized we had these cells, were very experienced. And so one of the things that Ningy wanted to do was to try and catch cells in the process of learning, but she never really managed. So it just seems like the moment you put the rat into the environment, suddenly it's expressing that bidirectional pattern. It seems to be quite quick. Of course, it takes a few minutes to build up the profile, because the rat has to have explored enough of the directional dwell, to be able for us to be able to see the pattern. So it may take a few seconds maybe to build up, but it's really quick as far as we can tell.
Okay. We have a question from Aaron Wilbur that apologized for coming late because he was teaching. And he asked that if the landmark to direction cells are similar to what he reported in PC, which is egocentric landmark, like and direction cells.
So this is possibly similar to Kevin's question, in as much as I don't think they're egocentric. I think they probably get projections from those cells, but I don't think they're egocentric, because the tuning curve is it's hard to rule out, because we haven't really tested this to destruction, but the tuning curve is there all over the box. So, and yet it's reasonably tight. It's a bit broader than a head direction cell tuning curve, but not much. It does seem to be kind of parallel across the environment, whereas the egocentric relationship to the rat of the visual scene varies as the rat moves around. So I don't think these are egocentric cells, but I think they're on the pathway from egocentric to allocentric. And maybe actually, if we tested this in a bigger environment where we could get more control of the relationship, we could unpick it. But no, I don't think they are in the simplest form.
Okay. We have a question from Mark, so you can just ask it.
Yeah, great talk, Kate. My question was actually about the learning process and how fast these cells develop if it's the first time, but you've already answered that maybe within the first couple of seconds, but you need sampling. But maybe a follow-up to that is in the other direction, from retrosplenial to or from the ring attractor to the retrosplenial, do you see sharpening of retrosplenial neurons in terms of their head direction coding in the open field with experience? Are they becoming more head direction modulated after repeated experience, even just in an open field?
We haven't really looked over time, I don't think. We could go back and do that. But the directional tuning that we saw, from those last few slides that I showed you, is quite broad anyway. Yeah. So it would be quite hard to see whether, subtle though it is, it's tighter. Yeah, but we'll go and take a look. Yeah, it's an interesting thought. They've picked up the direction.
Okay, and we will have just one last question, and after we can, the other can join on gather to discuss it. From Nora Newcomb, human neuroimaging studies also find the retrospinal cortex doing important work. How do we map from cellular to regional findings, or can't we?
Yeah, I've wondered about this, whether we might be able to see bidirectional cell activity. So it is possible, because now it's possible to do a sort of a multivoxel pattern analysis of the fine-grained activation. Problem in humans is that the retrosplenial cortex is quite small, but I think MVPA might be able, if you had a really strong magnet, might be able to show, for example, you could compare what happens in the bidirectional compartment between these, what am I trying to say? Between the two compartments in the bidirectional, bi-compartment box, you would expect some similarities, but also some differences, because some of your retrovenial cells remapped and some didn't. Whereas between that compartment and a completely different one, you'd expect a bigger difference because there was complete sort of remapping. So you might be able to see hints of this type of mixed processing in humans. But we haven't done that yet. But if someone wants to do that, that'd be awesome. Yeah, it's a good question.
Okay. Unfortunately, we'll not be able to answer all the questions now, but since it will be now the coffee break, I suggest you that we can continue the discussion on Gather Town, where you can just meet somewhere, there was spaces for everyone. And I also want to give an actual heads up that there is a treasure hunt that has been opened on Gather Town that has been created. So if you want to start it, you just have to go to the medular part of the main room, and if you don't know where it is, I'll just show it right now that, yeah, if you start to go there, you might start to see a treasure hunt. So yeah, be aware there is mazes and riddles. So yeah, good luck if anyone succeeds to reach the end. You know what he will be able to do, and yeah, good luck with that. And thanks again for all the talks and the sessions, and see you in 30 minutes for the next session, and see you on Gather.